Plant cells are made to fuse together under the microscope, controlled by an electrical charge. The presence of DNA, the blueprint of all living cells, is exposed under ultraviolet light. It's all part of a new industry, biotechnology.
Biotechnology is like microcomputers, telecommunications, one of the revolutionary new technologies that have come upon us in the last 10 to 20 years. They are going to radically change our lifestyle in the 21st century.
But the new industry is not as new as it seems. Using biological processes goes back thousands of years to early Egyptian times, when fermented barley was used to make beer. In beer making, the adding of yeast to a mash of barley, malt and hops causes the mixture to ferment. Microorganisms combine with the natural sugars in the mixture to produce alcohol. The new industry based on biotechnology is capable of producing a whole new range of products, new medicines, new foods, new sources of energy, new types of pollution control. And yet at the center of the new biotechnology is still the process of fermentation, but under very controlled conditions. What will transform the industry in the future is the ability to use genetic engineering to change the way cells grow, so that they'll produce things to order. In all cells, there's a nucleus which contains the genes. It's now possible for scientists to take the DNA from a gene in one cell and combine it with the DNA from a gene in another cell. If the combined DNA is then replaced, it will program the cell to produce whatever new substance is needed. So cells can become miniature factories. A scientific idea worked out in a laboratory is one thing, but how do you turn this knowledge into a profitable business? The leading British biotechnology company is Celltech. Here, work is being done to produce medical testing kits to enable doctors to identify viruses and diseases in the blood. They've already spent £5 million on research and development costs, but they're still not sure if their product will sell successfully. There's certainly no shortage of ideas at Celltech, but what will decide whether Celltech is successful as a commercial company is the ability to sell its products worldwide in a market which is very competitive. So in a high-risk business like biotechnology, what are the chances of a new company such as Celltech succeeding? We asked Peter Lang, who invests millions of pounds in biotechnology companies worldwide.
Celltech has an unusual access to some of the best of UK research. It's no secret to anyone that a lot of the ideas in biotechnology originated in UK universities, and the Americans have probably been better at exploiting those in commercial terms than we have ourselves, something that happens in many industries. In the USA, virtually every academic scientist has some relations with industry as a consultant, and he gets to know how industry thinks and what is required to make research ideas into worthwhile businesses. In the UK, that's comparatively rare, and as a result, academics don't really understand what industry demands, and equally, industry, which hasn't really called on the academics, does not appreciate what academics can bring them in new ideas about how things should be done.
Another recent entry into the biotechnology business is a company called IQ Bio, based in Cambridge. The company was formed to produce a new range of medical test kits which will help in the early detection of cancers and other diseases. The purpose of one of their tests is to produce a heightened colour reaction which will measure the presence of antibodies in the bloodstream caused by disease. But will this high technology process find a place in a very competitive market?
Well, it's certainly a growing market, and it's certainly becoming a more and more competitive market. We obviously hope that the clear-cut benefits of our system will be an important factor in selling the kits and the whole technology. We also recognize that as a small company, we will have to grow very quickly to cope with this.
If a company decides to expand, it needs to borrow money. And a new company like IQ Bio would have to approach a bank or other companies with money to invest. One such investor is Peter Lang.
We only have a limited sum of money in which we have available to invest. If we invest in a bad business venture and we lose our money, then that money, by definition, is not available for the good ventures in the future. Now, the criteria we lay down demand that companies have a good team of people, they have the right business approach, that they have the right technical skills, the right managerial skills, and perhaps more as important as anything else, the drive and ambition to make the company succeed.
So when money is invested in a company like this, where does it all go?
The prime investment is in the people that are spending the time doing it, because they cost a lot of money. On the average, a scientist costs between £30,000 and £50,000 a year by the time you've paid his salary and given him something to work with and a place to work. What you have to do after you have the idea and proven it in the laboratory is you have to scale it up into production so you can make enough to test. When you've got enough to test, then you have to go out into the field, and whether it's testing it on plants or on animals or on patients in a hospital or on consumers, if it's a new cornflake for that matter, you can spend a great deal of time and a great deal of money in doing the scaling up and the testing without any guarantee that you're going to have a successful product when you're finished.
A product which illustrates both the potential and the risk in biotechnology is interferon. Hailed originally by the media as the new wonder drug which could cure cancer, interferon didn't live up to its promise. The research on tumours in mice was encouraging, and interferon seemed to have a measurable effect in reducing the size of the tumours. But when the drug was tested on humans, there was no evidence that it was capable of curing cancer, certainly not in an advanced stage. The investment in interferon worldwide has been colossal, and there are now over 70 major companies working in this field. One of the leaders in producing interferon is the Wellcome Foundation, with one of the biggest plants in the world. But until there's more evidence for the success of interferon in treating viruses or tumours, it's impossible to know if all this investment is justified. Producing interferon could be the greatest money spinner in the history of the pharmaceutical industry, or one of the biggest disappointments. We asked John Walker of Investors in Industry for his view.
Extensive trials have been taking place with a number of interferons. There aren't just one, there are a number. The trials, by and large, it isn't the magic bullet that cures cancer, cures anything. What people are now coming to realize is that there is a probability that in association with other products, it may have a useful component. It is nothing magic. The fact is, though, that the amount of effort and research that went into interferon has done one thing that's very important. It's advanced the technology. But as a commercial product, I certainly wouldn't put any money behind it.
Another major company that's invested in biotechnology is the chemical giant ICI. For over 14 years, they've been researching new products, one of them right through from the laboratory stage and the pilot trials up to full-scale production. The idea behind the project was to create an animal foodstuff that was rich in protein. This could then be a substitute for the other forms of protein obtained from natural sources like soybean and fish meal. All the animal trials with the new protein were successful, The trials confirmed that animals liked it, it was nutritious, and it improved the animal's growth rate. From the trials, there appeared to be no side effects. The next stage after the animal trials was to build a massive fermentation plant that would produce the protein in large quantities. The fermenter had to be a completely sterile environment to grow a specially selected bacterium which fed on methanol. But in spite of this huge investment, Protein has not been a commercial success. What ICI planners were unable to predict was that the price of methanol would rise faster than the price of the soybean product they were competing with. But the experience gained in creating a large-scale fermentation process can be put to other uses. In this fermenter hall, ICI is growing a bacterium which produces a new form of plastic. It's made by a natural bug, not a genetically engineered one. In this photograph, the bacterium has a string of the plastic spilling out of it. One of the problems was to find a way of splitting open the bacterium to release the plastic. When the fermentation process is over, the newly produced plastic is dried into a powder. In this pilot plant, they're experimenting with ways of converting the powder into different forms. One way of producing it is in the form of pellets. Another method is to squeeze it out into a long string. What this plastic has over other plastics is that it will eventually rot away. It's biodegradable. And that property has fired the imagination of scientists at ICI into thinking up all kinds of new applications.
About the time we were thinking of use for this plastic, Barry Sheene had his celebrated motor accident when he was badly injured, and X-ray photographs of his leg appeared on TV showing lots and lots of stainless steel pins. And we thought that if you can take something like our biodegradable plastic, which is still very stiff, very tough, very strong, and replace those stainless steel pins with these plastic pins, they'll degrade away slowly in time. They'll leave a stronger repair, and they won't require a second operation to remove them.
A plastic like this, which will dissolve away, has many advantages over conventional plastics, which do create a serious waste disposal problem. ICI have been successful at making the product, but as yet they've no guarantee that they'll be successful in selling it. Biotechnology can now be used to control the growth of bacteria in ideal laboratory conditions. But could this knowledge be used against insects which cause disease, like these mosquitoes, which can be carriers of malaria? Could one form of bug be used to attack another one in a kind of biological warfare? That's what scientists in Tate & Lyle's research department decided to try and find out. They identified the microorganisms which will attack mosquitoes when they're in the larvae stage, and they found a way of manufacturing them. When the bacteria are released, they're eaten by the hungry larvae. Once inside, they cause the larvae's stomachs to blow up, quickly killing the insects. An insect that causes damage to plants grown in greenhouses is the aphid. To deal with aphids, a different kind of insecticide was developed based on a lethal fungus, which when sprayed onto the insects would grow on them and be transmitted to other aphids and infect them. Aphids have become very resistant to ordinary insecticides, but the fungus is not an ordinary insecticide. The fungus is eventually seen as a white growth completely covering the aphid. Minute fragments of pea plants grow in a tissue culture. Light and temperature are controlled. The atmosphere is free from contamination. A superior strain of pea is being cloned in ideal conditions. It sounds like Brave New World, but it's happening now. If you think of an ordinary pea plant as a miniature pea factory, it could be more efficient in producing peas if it had fewer leaves. If a scientist were to redesign this plant, it would have hardly any leaves at all. By normal methods of crossbreeding, it would take years to achieve this effect. But scientists are now working on a method of transferring directly the genes of one strain of plant into another one by fusing two cells together. The culture is placed in an electrical field, causing the cells to form into chains. A high-voltage pulse causes some of the cells to fuse together. In time, it may be possible for scientists to transfer genetic material between the cells from two distinct species, but this type of genetic engineering in plants has still a long way to go. Another technique being practiced at the John Innes Institute is to use a minute needle carefully controlled through a microscope and to attempt to inject the cell with a substance. If a technique like this is successful, it would make possible the direct injection of DNA into a single cell so as to change its genetic characteristics. And from that single engineered cell, thousands of clones could be created. It would be hard to imagine a role for biotechnology in ordinary domestic refuse. But there are bugs in rubbish, and one man has thought out an ingenious way of making them work for him. How can all that waste be turned into something useful? That was the problem Vic Lawson set himself. The first thing to do was to separate the main components of the rubbish. The secret of this part of the operation lies in a huge revolving drum which sorts the rubbish out. Inside the drum are specially designed filters which allow the vegetable waste to be isolated from the other waste products. These are then taken away for recycling or disposal. The vegetable matter is filtered out from another part of the revolving drum, neatly shredded to make it ready for the next part of the process. From this point, the vegetable matter is taken off to the fermentation tank, where it will be combined with other substances. The precise chemical formula of the mix is a closely guarded secret, but there's no attempt to conceal one of the main ingredients: liquid sewage. As the product of the fermentation emerges, it looks as if only a gardener could think of a practical use for it. But this is just the raw material. Vic Lawson has found a way of turning it into marketable products. A material like peat. A wood fibre. An artificial fuel. And chipboard.
Everyone has a waste disposal problem. With this sort of process, you solve that problem. You take away health hazards, you take away smells from the society, and in turn you give them back resources that are very important to the society.
So, from the unlikely elements of domestic rubbish and liquid sewage, Vic Lawson has invented a way of creating raw materials which people can use. He's proved that his process works, and maybe he's created the growth industry of the future.
Every single developed country will take this. I've been to Australia, I've been to Hong Kong, I've been to Norway. Everywhere I've been, the response has been positive and definite, and I'm certain we will sell plants in all those locations and many more.
WMC is essentially an individual entrepreneur who had an idea, but instead of rushing straight to financiers and wanting money for it, spent a lot of time and a lot of his own money in developing the project and the idea into such a form that it was suitable for financing. And he's quietly and competently built up a team of people. And that's just the sort of approach we like to see, and it's one that is so rare in this country.
In the high technology business, we need scientists. We need their ideas. It is the lifeblood of the industry. Conventional business doesn't need them. We do. And therefore, if we have to beat a path to their door, if we have to find out what the marketplace is for their products, we have to introduce entrepreneurs for marketing, selling and production, so be it, that's what we will do. But it is a very high-risk business, highly competitive, and it's also a worldwide business. You can't do high technology within national boundaries. You compete on an international basis. And therefore, we have got to ascertain that the technology in Britain is the best. If it isn't the best, it's not worth doing.
In just under 5 minutes, Hold Down a Chord, folk guitar for beginners.